Good sleep habits. And the waking up and sleeping at the same time is very appropriate. Even on the weekends, you should try to still wake up at an appropriate time, go to bed at an appropriate time, and you'll notice your rest will become much better. It's also important for forming long-term memory. So people always like students, they always want to cram the night before the test and study and study and study and stay up late and study. Our brains don't work that way. The way long-term memory is formed is as you sleep, the brain does the mapping then. So when you study and you sleep at an appropriate time, it actually locks it in. So you get more bang for your buck from your studying. So it's a good habit to get into. And I always tell students, my best studying as when I was a student was in the morning. While I'm having like my cup of coffee or something, study then, like fresh material, like really like get into it then. And then you go to school, whatever, work, and in the evening, you're kind of doing review work because your brain's already tired. In the morning, it's fresh. So steer clear of melatonin, all right? If you really need help sleeping, a better choice is Benadryl. Benadryl is much quicker acting. That's what's in ZQuil, okay? That's all it is. It's Benadryl, overpriced Benadryl. But that can be taken, it's non-habit forming, and that can help it with you adjusting like sleep cycles, stuff like that. Right. All right. So as far as melatonin goes, there's also this whole like initiation of reproductive cycle, so it helps with that sexual maturation. So low melatonin can have an effect on that. So we form melatonin, if I remember right, it's from tryptophan, the amino acid tryptophan, and certain foods are very high in tryptophan, like turkey and milk, and it converts it into melatonin in our body. So that's why, like after Thanksgiving dinner, everyone just wants to go to sleep, right? That's in part why. Thymus, so located in the mediastinum. As a child, it's much larger. It plays a role in our formation of our T cells. So thymosin is what actually stimulates the T cells and like develops them, trains them, all of that. So thymosin is the um, protein that is doing that. Go through these guys. We looked at them throughout the semester, so run through them. Make sure you're familiar with the different hormones. Right? Estrogen, so we're going to talk about that in reproductive. The big production of estrogen is the granulosa cells around the egg that they are the ones that are producing the majority of the estrogen. So that's why when a woman runs out of her eggs, estrogen levels drop. Yeah. And then progesterone, this comes from corpus luteum. So after the egg is ejected out of the ovary, we have what is called corpus luteum that makes progesterone. Now, if the egg implants, if it's fertilized and it implants, it'll continue to produce estrogen. The estrogen keeps corpus luteum alive, so we keep progesterone. Both keep the uterus lining from shedding and maintaining the pregnancy. And then testosterone in the males, that is produced by the Leydig or interstitial cells, so between the seminiferous tubules, and we need that for sperm production. Purely endocrine glands, just a little reminder for each of those. And then endocrine and exocrine. All right, stress. So we kind of saw this with shock. It's kind of the same concept, how we lay these out. So, which phase are you in? Alarm phase. So here the system's coming online and it's upregulating everything. So this is immediately after stimulation from the sympathetic system. So we see the increased heart rate, blood pressure increasing, glycogenolysis, lipolysis, proteolysis, increased ATP production. All right, we are in full like run phase. Whereas resistance phase, this is where we go on for an extended period of time. When this happens, we start to release cortisol. Cortisol is still going to do a lot of the things we were just talking about, that increase the glucose level, increase the blood pressure. The downside here is that immune suppression. So we see this for when the body is in an extended phase of, stress. So here they give examples like starvation, acute illness. So this is like 2 weeks or less, severe anxiety, stress, emotional stress. And we can see that cortisol increase. And a point on here is we will conserve glucose. Why do we conserve glucose? Because the brain needs it. It can't run without it. So it'll try and conserve glucose for the brain. And then exhaustion phase. The problem here, we're running out of resources. We've stayed in this, elevated abuse of our body for an extended period of time. And so we start to see the cortisol levels cannot be maintained. And now we've burned through fat on the body. We start attacking the organs. We're pulling protein out of muscle, out of the organs, everywhere. And so we start to see electrolyte imbalances. This is end of time kind of thing. This is when the body is shutting down. Okay. All right. Now we're going to move on to reproductive. Let me see if this is the one I want to use. Yeah. So this one is a little longer. Don't freak out by the number of slides. The reason is it's going to go through meiosis in detail and show you each step in an image. So that's why it beefs up the size of the PowerPoint, but in reality, it's not anything wild, okay? So meiosis, we're going to talk about, that is where we're taking and forming sperm and the egg. And we're taking, in the human body, we're taking a cell that has 46 chromosomes and dividing it down to 23 chromosomes in each cell, and they are unique in each cell. They're unique from each other. Oh, and even the DNA is unique from the parent cell. All right. Gametes, that's sperm and egg. Gonads, that's what generates them, so ovary and testes. Chromatin and chromosomes. Chromatin is when your DNA is all spread out and you can read like the A, T, G, C on there, versus chromosomes, it's condensed down. So what we'll see is we condense the chromatin, this long spread out DNA, down to chromosomes so that when we duplicate it, we can split it easily and evenly. So human cells, 46 chromosomes, referred to as diploid, meaning it has both pairs of chromosomes from each parent. So we'll say there's 23 pairs of chromosomes and they're homologous chromosomes, meaning you have a chromosome 1 from mom, a chromosome 1 from dad, a chromosome 2 from mom, a chromosome 2 for dad. They code for the same things, but in reality, use one or the other or parts from each chromosome, not necessarily both equally, okay? So that's why we see we're like a blend of the parents. So diploid has both sets of DNA, both from each parent, versus haploid. So haploid is when we start to talk about either the sperm or the egg. They have half the DNA. So what happens when we take the two halves, we combine it, we fertilize the egg, it becomes a zygote. That now one cell, we took two cells and made one cell, and it now has 46 chromosomes. Is that not spectacular math? We took two cells and turned it into one cell. 1 plus 1 equals 1. Also, 1 plus 1 equals 3. Man, woman, 3 people in the end. Bet you didn't know this math. What's the new Common Core math that they do? That's what this is. All right. So, fertilization, we take the 2 haploid cells, the egg and the sperm, we combine them, it becomes the zygote. It is one cell with 46 chromosomes in it. It has each chromosome, like each number of chromosome, 1, 2, 3, 4, one from each parent. Okay? It now is a diploid cell. The zygote is considered diploid. What's wild when you think about that, we start off as one cell and it has all your DNA in it. It divides and differentiates into all the unique cells in your body. And how many times it multiplies. You think about it, in 9 months, you go from 1 cell to 7 pounds or something. That's pretty impressive. So all the chromosomes are considered homologous except for sex chromosomes because these can be different. They code for different areas. So female is XX, male is XY. So meiosis, we're going to talk about oogenesis or oogenesis, that's making the egg, and spermatogenesis, that is making the sperm. So we start with 1 diploid cell and we end with 4 haploid cells. Now, in reality, that is true for the male. Females, you actually make 1. The reason being you steal the cytoplasm to supply a much larger cell, and so the others aren't viable as it divides. But they say 4. We can talk about 4. So meiosis is like mitosis times 2, but we only go through S phase of interphase where we duplicate the DNA once. We do it in the very beginning. Then we go through meiosis 1, we divide the cell. They technically have 46 chromosomes in each one. Then we go through meiosis 2, we skip S phase of interphase, and we'll divide those 2 cells now into 4 cells with 23 chromosomes in each one. The reason, even though in meiosis 2, they have 46 chromosomes in there, it is considered haploid because we separate mom and dad's DNA. And so you're missing some of the DNA. It's considered half the DNA in there. So whenever you guys are looking at this, you have to tell me, like, if there's a question about, like, what phase are we in? You can't just put metaphase. You have to tell me metaphase 1 or metaphase 2, meiosis 1 or meiosis 2, because the events that are happening in each is different. And so you need to know what's happening in each step. So usually what I tell students is, like, draw it out. That's one of the best ways. Draw it out on a piece of paper, flashcards, and then have what's happening in each step. That's the easiest way to learn it. So at the end of meiosis 1, technically started meiosis 2, we have 2 haploid cells. They do have 46 chromosomes in each, but they are considered duplicated chromosomes. What that means is the 2 chromosomes are stuck to each other. Okay? So here's a regular cell. These are just individual chromosomes. Okay? This is after S phase of interphase. We've duplicated the chromosomes. In prophase 1, one of the big things we talk about is the nuclear envelope disappears. And then this is another, like, crucial point that happens. So what's happened here is chromosome 1 from mom, chromosome 1 from dad line up next to each other. Chromosome 2, 3, 3, 4, 4, and so on, okay? They all line up next to each other. And these are called the tetrads. The reason they're called tetrads is this half is a sister chromatid, sister chromatid, sister chromatid, sister chromatid. There are 4 structures lined up next to each other. What's going to happen here is crossing over or synapsis, and we share the DNA. So like in your body, your mom and dad's DNA are going to swap with each other, little pieces. Where it happens is different every time. So doing this creates genetic variability. Okay, so this is crossing over. And that's why no matter how many offspring you have with the same partner, they're always going to be different. The DNA will never ever be the same because where this happens is unique every time. Okay? So this creates genetic variability for us. Metaphase 1, the tetrads line up in the middle. Anaphase 1, we separate the tetrads. After telophase 1, cytokinesis 1, we now have 2 daughter cells. They're unique. They're haploid. They have 46 chromosomes in them, okay, or 46 sister chromatids in them. Technically, it's 23 chromosomes. It's such a technicality here. This is considered one chromosome, but the second we split this apart, it's 2 chromosomes. So technically, there are 23 chromosomes in each cell or 46 sister chromatids. So at the end of meiosis 1, when we start meiosis 2, we have 2 haploid cells. They have the 23 duplicated chromosomes in each. And we're going to separate this in meiosis 2. So we go through basically the same thing. We do not duplicate the DNA. So no S phase of interphase. We go straight into prophase 2. I don't even know that the nuclear envelope redevelops or to go away again. It should just jump it. Metaphase 2, who lines up in the middle? The sister chromatids or chromosomes. Duplicated chromosomes, okay? Then anaphase 2, we separate the sister chromatids or the duplicated chromosomes. So now there are 46 chromosomes in each cell or 46 chromatids. They're no longer sister. They're not stuck to each other. Then we go through cytokinesis, telophase 2, and we have 4 daughter cells with half the DNA. So there's 23 chromosomes in each one. Each are unique from each other. Each are unique from the parent cell. So end result, 4 haploid cells with 23 chromosomes in each one. So, like we said, meiosis, we start with 1 diploid cell. After we complete meiosis, 4 haploid cells that are unique from each other, unique from the parent. 23 chromosomes in each one. So this image is nice because it's comparing the two. So you can see here when we go through with the sperm, we end up with 4 cells. Here, so the males are doing this from start of puberty till the day they die. And this, every 2 months, they complete the cycle. Like the sperm will complete the cycle, mature, and be ready. Females, they are born with their eggs, and they are arrested or held in prophase 1 until puberty. Then once the body realizes, okay, it is mature, it can handle a child, FSH will be released and will start to stimulate those primordial follicles. You should have looked in lab at last week, okay? And they will start to mature. It takes about 2 weeks for this process. As they do that, we'll create what are called polar bodies. So what's happening is it's stealing the cytoplasm and giving it to this survivable cell so that it is larger. And it needs that to accommodate the DNA coming in from the sperm. So as it matures, we'll form 3 polar bodies and 1 true egg or ovum. Ovum is an unfertilized egg. Once the egg is fertilized, it's zygote. So spermatogenesis, that's making spermatids, sperm. Okay. Spermiogenesis is then making sperm from the spermatids. So initially we make the spermatids, and then as they mature, they become sperm. So the base stem cell in the male is going to be spermatogonia. Spermatogonia, they can undergo mitosis, they can duplicate themselves, and then from there, they can become sperm. So when they start to undergo meiosis, then they're going to be converted into the four spermatids or sperm cells. So the first one we look at is the primary spermatocyte. So this is that initial cell. So whenever you guys are looking at this in lab, you kind of start on the edge, and the real dense structures, that's the spermatogonia. As you move in towards the center of the seminiferous tubules, you're jumping through the different spermatocytes, okay? So when the primary spermatocyte looks kind of dense also, the secondary, you don't see as much stuff inside of it because they have half the DNA. We've separated the DNA. So as it continues on, it'll become the four spermatids once it divides again, and then it'll undergo spermiogenesis for the next few weeks, so a total time of about 2 months to mature. And then it'll be viable and ready, to fertilize an egg. So when we have the spermatid and we undergo spermiogenesis, what's happening is it's forming this acrosomal cap, which protects it, and then forming like its tail, the flagella, tons of mitochondria in here so that it can move. When we look at like what makes up semen, we'll see like there's fructose in there for energy, sugar so it can generate ATP so that it can swim. We just looked at this. Okay. The female, when they're born, there's no true like stem cell. It's gone. So the equivalent of the spermatogonia is gone, which would be the oogonia or oogonia. Instead, they're born with the cell, like we said, arrested in prophase 1. It's held there until FSH comes along. When FSH comes along, then it'll start to process through the rest of meiosis. So as it goes on, we'll go from a diploid cell into the secondary oocyte, which is haploid. So that means we've completed meiosis 1. We are in meiosis 2. There's that unequal division of the cytoplasm. It is stealing the cytoplasm to enlarge the cell. So the other split, the other half after we divided, kind of phases out. That's the polar body. It should not be able to be fertilized or continue on. So as it's processing and it's maturing, so we're going to see it goes from primordial to primary to secondary to Graafian or tertiary follicle. And then what we'll see is at this point, there's tons of estrogen being produced. The estrogen stimulates the mind or the brain to release luteinizing hormone or LH. That LH surge causes the egg to be ovulated. It is released from the ovary. It will continue now. It's at metaphase 2. It will continue through meiosis if it is fertilized. Okay? If it is fertilized, it will mature. It is going to then implant, and we will have a fetus. Okay? So if it's fertilized, then it's a zygote. It'll continue on, creating another polar body as it divides, and then implant. Okay? So here, this is what we were just running through, up through ovulation. Right? So that's what they're showing here. And there's the rest of that follicle that becomes corpus luteum that makes progesterone. These cells right here, these granulosa cells right here, that's what's producing estrogen. So really, it's all these cells. They also call them like follicular cells. You hear both. You'll hear granulosa, follicular cells, one and the same. Uh, they produce estrogen and they also produce progesterone, but when it becomes corpus luteum, it produces more progesterone than estrogen. Okay. But corpus luteum also produces both. It's just the way these notes are written or the book's written. It wants to say one side is heavy estrogen, the other side is heavy progesterone. So males, they have the spermatogonia, that base stem cell, and that's why they don't stop producing sperm till the day they die. Whereas females, because there's no oogonia, they run out. When they run out, that's menopause. Okay. And the biggest indicator of when a female will have menopause is the age her mom had it at. We say 50 years plus or minus 2 years, but really it's mom's menopause age is the most accurate. Okay. All right. Reproductive system. So you guys in lab, you'll be looking at, the models, stuff like that. But we've got ovary, we have fallopian tubes, uterus, and then the vaginal tract. So a couple things here. We see there are structures that are homologous. They are equal to the other sex. So the ovary, its equivalent is the testes. So a couple things here. One, there's a couple different layers or ligaments, attachment points that we need to know. You need to know like directions where it's attaching. All right. So tunica albuginea. So that's just the capsule that is surrounding it. Cortex, just like the kidney, is the edge. And then medulla is the center. So on the cortex, you'll find the primordial follicles. And then what happens is they migrate into the medulla and they mature. And then they migrate out to the cortex and they spit out what is called corona radiata. It's the egg with the granulosa cells around it. Those cells, if it's fertilized, the granulosa cells or follicular cells will become the placenta. That's what that becomes. Okay. Ligaments attaching the ovary. So we have the ovarian ligament. So this is ovary to uterus. All right. So I want to click. Ovary to uterus. So it's this guy right here. Okay. And then we have suspensory ligament. This is ovary to lateral pelvic wall. So to the side. We can't see it here technically, but it would be going out to the side there. So they have it. They have ovarian and suspensory here. The suspensory carries vessels and nerves. Right? So controlling the ovary, the hypothalamus. The hypothalamus is always in control of everything, right? So he releases the releasing hormone. That goes to the pituitary. The pituitary then releases FSH. That stimulates the primordial follicle. This happens really at somewhere in the menstrual cycle is where this will start to happen. The menstrual cycle will happen, we'll release FSH, we'll start to train that primordial or develop it. And then when we reach this ovulation point, that is 2 weeks before menses. That point usually lines up pretty darn accurate. Okay. When the egg is ovulated out, that's because there's this LH surge due to a high estrogen volume. So this now can be fertilized. So a big common misconception with people is like they think you can only get pregnant at technically on day of ovulation. Not true. It is something like 3 days before, 3 days after. So almost a full week the egg can be fertilized because the sperm can survive in the fallopian tube for a certain amount of time. And then the egg can also survive and not be sent out immediately and also maybe released early. So it ends up being roughly a 6-day window where pregnancy can happen. So we're going to look at two cycles, like ovarian and uterine. They go together. The events go together because they are stimulating each other. Really, the egg is what's controlling all of this. Okay. So the primordial will be stimulated and become the primary follicle. So inside of the primordial is the primary oocyte, the true egg. All right. So that primordial follicle, it's shielding. The reason we have these cells around the primary oocyte is to shield the immune system from seeing it. If the immune system sees it, and you saw how we just went through meiosis, that DNA is foreign to your body. It is mutated and it's missing DNA. Okay, so it only has 23 chromosomes in it and it's different. So if your immune system sees it and identifies it, it will attack it. So that's why you really have the follicular cells around it. That's one of the biggest reasons. So at puberty, the body will reach a point where it can handle a pregnancy. FSH will be released. The woman will ovulate. If it is not fertilized, she will have menarche, her first period. A woman can get pregnant before her first period. Okay? And I think I was telling my lab students last week, we were talking about this. It happened, my preceptor in peds, she had a case like that. The mom brought in the child. She was having odd weight gain. And the second the pediatrician laid the girl down, she could tell she was pregnant. Right? I had to explain that to her mom and didn't believe it. And she was like, there's no way. She's never had her first period. She already ovulated. Okay? She was sexually active before then. They had to go down that whole spiral of not fun. Okay. So yeah, the ovarian cycle will kick in at different points for women. Okay, menarche. We're seeing it earlier and earlier, and they keep on. You hear people. It's the antibiotics and the hormones in food. It's the high nutritional value of food. The body's growing at peak pace, if you will. I mean, you look at little kids now; they are either not either, but you see a lot of obese kids, and then you see like they're like little monsters in elementary school and middle school. Like they're tall, like they're just growing at a better pace because we have lots of nutrition in our food. The calorie intake is huge. All right. So the body accelerates its growth rate at that point because you're feeding it. All right. So the movement of the egg, the maturing of the egg is the ovarian cycle. The maturing and shedding of the uterus lining, that's the uterine. So what we'll see with the ovarian cycle, the follicular phase, this is the 1 to 14 day. This one isn't always super accurate. Okay. Ovulation should be on the 14th day. The luteal phase is pretty darn accurate. Okay. So this one, when a woman ovulates, she can project when she'll have her menstrual cycle. Sorry, I'm reversing that. The follicular phase is very accurate. It's the luteal phase that is not. Right? Yeah. Yeah. All right. So as far as follicular development goes, and we're going to look at each of this, that primordial is going to change from simple squamous cells to cuboidal, double them, triple them, create an antrum, get bigger and bigger and bigger, shift the egg down to the base, and that'll be the Graafian follicle or tertiary follicle. That's really where we'll see tons and tons of these granulosa cells, and that's where we see that estrogen climbing and climbing. Okay. Difference between a primary and secondary follicle, an antrum. A gap between the cells with fluid in it. That's how you know the difference. If you don't see that fluid space, you're still in a primary follicle. So you'll see cuboidal cells, and then you won't see like a secondary space. When you see that secondary space, you're now in a secondary. The tertiary follicle, it always has that big antrum, and the egg is positioned on one side. It's not central, it's always pushed to one side. And this is where we're going to see tons and tons of estrogen being produced. So when that high estrogen comes through, it stimulates the brain to release luteinizing hormone. Luteinizing hormone causes ovulation. What is ovulated? What is sent out? It's called corona radiata. It is the egg with granulosa cells around it. That now will be picked up by the fimbriae, the little fingers hanging off the fallopian tube, and pull it into the fallopian tube. And then it'll be fertilized potentially. Okay? It's fertilized in the ampulla of the fallopian tube. The remaining part of the Graafian follicle, those follicular granulosa cells that are left behind, will involate on themselves, they'll collapse in on themselves and form corpus luteum. Corpus luteum will produce progesterone. It'll stimulate the uterus to engorge and grow even more to accept that fertilized zygote if it exists. So, primordial follicle, simple squamous, okay? Primary follicle, we start to see cuboidal cells, and this is happening because FSH has been released. So here the follicular granulosa cells are producing estrogen. They're also producing progesterone, but estrogen, okay? So as they mature and we get multiple layers and we get a gap, that's that antrum. Now we're a secondary. So we're still continuing on, and eventually we hit Graafian. There's the egg wedged down here. Now we have the highest estrogen amount. This stimulates the brain to release luteinizing hormone, and that in turn stimulates him to be ovulated. So here, FSH comes down, we go to primary, there's the antrum, there's the Graafian. Estrogen is incredibly high here. Now LH comes down, and we release corona radiata. This remaining structure becomes corpus luteum, and then eventually down the road, about 2 months or so, it becomes corpus albicans. It doesn't do anything. So, days 1 through 14, follicular phase. It's the follicle phase. Estrogen climbs, 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 climbs. Eventually, it causes the LH to be released. When LH is released, now corpus luteum is active, hence luteal phase. Here we see progesterone levels increasing and possibly fertilization of the egg and implantation. If not, rinse and repeat. We'll just do the same thing over, okay? We'll keep on going through that cycle till we run out of eggs. So FSH stimulates the follicle. The follicles make the estrogen. The estrogen makes the brain release luteinizing hormone. Luteinizing hormone causes the ovulation. The tertiary follicle or Graafian follicle, that's where we're seeing estrogen and progesterone coming from. That's what's in turn causing this. So it's kind of like a domino effect, all right? So in women, when they want to say preserve their eggs, this is exactly what we do to them. We give them FSH injections. They take that, and then on the 14th day, we give them LH, and then we harvest the eggs, we capture them, and then they can store them. They can have them fertilized, reimplanted, whatever they want with them. They can donate them. They're very expensive. I think you get something like 10 grand for each donation. Something crazy like that. My wife needs to donate some eggs. Just kidding. She might beat me. We have a mortgage, okay? All right. So the follicular phase, like we said, estrogen levels are slowly rising, and eventually we hit a peak, and that causes LH to be secreted very rapidly, hence the surge. That causes the ovulation, and corpus luteum and the granulosa cells around corona radiata produce estrogen, and then also corpus luteum, the big thing is progesterone. So this image is actually really neat for this. So here's FSH. There's the primordial developing. As it develops and we get this a large amount of estrogen, that triggers the LH surge. There's the ovulation. Now we form corpus luteum. So you see estrogen levels can be maintained and progesterone ramps up. Eventually collapses and becomes corpus albicans. Now, simultaneously, what we'll see is estrogen makes the uterus lining grow. That's the proliferative phase. And if progesterone comes out, secretory phase, it engorges. It's full of glycogen, ready to accept the zygote to implant. And if it doesn't, we see the egg leaves the body. The progesterone falls off because there's no estrogen to stimulate corpus luteum. So it becomes corpus albicans. Nothing to maintain the uterine lining. It sheds. Menstruation. And we do it all over again. All right. Fallopian tubes. So, as far as the fallopian tubes go, they're not connected to the ovary. They have these little fimbriae, these little finger-like structures. Then we have the infundibulum, the ampulla, that's usually where we see fertilization happen, isthmus, and then open into the uterus. It's intraperitoneal, nothing else there. So it has ciliated cells. The cilia is needed to move the egg along, as well as the mucus in there. The cilia become damaged from pelvic inflammatory disease, and that's caused by chlamydia, gonorrhea, so STDs. So STDs have to be treated. The problem is with some of these, like women aren't symptomatic sometimes, and it climbs up into the fallopian tubes and destroys the cilia, and they become scar tissue. If it becomes scar tissue, the egg gets stuck. There's no cilia there to move it along, and the egg gets stuck, and then it gets fertilized. And that's how you end up with a fallopian tube ectopic pregnancy. Destroys the fallopian tube. You end up with just one functional side, and depending on the egg to release from that side. Right? There is smooth muscle that helps through peristalsis to move it, and then the serosa. You have the mesosalpinx, which is the ligament that is covering it. Okay, so broad ligament going over it and covering it. So fertilization, like we said, happens somewhere in the ampulla roughly, and then the cilia, the smooth muscle, the mucus moves it along. When it implants, we call it the blastocyst. It is no longer a zygote. Okay? All right, uterus, endometrium. So stratum functionalis, that's the portion that builds up and sheds during menstruation. So it's the functional layer. Stratum basalis, this is near the myometrium, the muscle, and that's where it generates the layers and pushes it up, okay, to regenerate functionalis. Myometrium, smooth muscle, nothing special there. And then has perimetrium, which is the visceral peritoneum surrounding it or stuck to it. So this is the free layer. So all the way down to through here would be endometrial. Stratum functionalis just somewhere in the top here. Right? There's really no dividing line. Stratum basalis would be at the base, and then there's myometrium. These are the endometrial glands, all of these structures that you see in here. Oops. All right, so as far as the uterus goes, it is anti-flexed. So it's pushed forward. That's what that means. Um, Sometimes they sit a little more vertical, they sit a little differently. Just depends woman to woman. Uh, the round ligament, so this is what's holding it anti-flexed. This is the one that hurts typically when women are pregnant because the pulling on the anterior wall or ventral wall of the body. Uteral sacral ligament, so this is between uterus and sacrum. And then broad ligament, so we'll see he has three parts to him. Mesometrium, that's over the uterus itself. The mesosalpinx we just talked about over the fallopian, and the mesoovarium over the ovaries. So that makes them intraperitoneal since they are covered in it, encased in it, right? All right, so the uterine cycle or menstrual cycle, it's going to overlap with the ovarian. So hypothalamus is controlling it. So when the hypothalamus releases FSH, we start to stimulate that primordial follicle, we see estrogen levels. Estrogen makes the uterus grow. Then we see progesterone levels from the corpus luteum. That makes it grow even more rapidly and engorges it. So we see lots of glycogen, blood in there, and it's getting ready to mate with the placenta and provide it with nutrition. If the egg is not fertilized, it leaves the body. Well, there goes the estrogen. If the estrogen leaves the body, the corpus luteum is not stimulated, the progesterone decreases. When they both decrease, that's it, menstrual cycle. We shed that endometrial layer, the stratum functionalis. So, two are connected through hormones, like we just said. So, the proliferative phase, this is the growing phase. It's proliferating. That's stimulated by estrogen. Estrogen is starting that after a menstrual cycle. So it's going to occur at the same time as the follicular phase because we see estrogen ramping up. So as it continues on, the endometrium grows and then progesterone comes in and really ramps it up. Okay, so the progesterone is due to corpus luteum. So that is what is causing the luteal phase, right? That's why we call it the luteal phase. So if the egg becomes fertilized, it becomes a zygote and implants, it becomes a blastocyst, then we'll maintain estrogen, we'll maintain progesterone, we'll maintain the uterine lining, and the pregnancy will continue on. So this is the secretory phase, and that's really due to progesterone. So you can see the endometrial glands become like huge. So fertilization, like we said, ampoule of the fallopian tube is usually where it occurs. And once it is fertilized, it is called a zygote. And progesterone. So progesterone increases body temperature, so can estrogen. Uh, and that's why women can do like temperature logs or diaries, and they can figure out when they are ovulating that way. Or there's an LH test kit. You just urinate on, you pee on a stick, and it'll tell you if you're ovulating or not. Okay. The Progesterone from corpus luteum is keeping the uterus lining active. Eventually, the estrogen, the granulosa cells turn over and start producing beta HCG. Beta HCG is what we use to test for if someone's pregnant. It's in the urine. Um, so it is a growth hormone, choreotropic growth hormone, that is secreted by the placenta. So that is a way to tell if someone's pregnant. It's also what is monitored after, like if someone has fetal demise, right? They miscarriage, something like that. That's what we monitor to verify that all the placenta has left the body. Um, After giving like abortifactants. So human chorionic gonadotropin, so that's what's eventually going to maintain corpus luteum, and that's the pregnancy test. Males can test positive for beta HCG. It means they have what's called an egg yolk sac tumor. It's a tumor of the testicles. So you may have heard stories. They talk about like, oh, my boyfriend peed on the pregnancy test and it turned positive. Yeah, take him to the doctor. Right. All right. So, menstrual cycle, menses. If there's no fertilization, that means there's no implantation. There's no maintaining progesterone, estrogen, or beta HCG. That's it. It falls off, and we start to shed stratum functionalis. And we have menses. And because there's no progesterone, there's no estrogen, who starts back up? FSH. FSH will start, and we'll just go right through that cycle again. So this is a good little overview chart as well. And then it actually includes, like, you can see not the hormonal a hormonal change that causes the temperature change, right? So what women will do is they can just log their daily temperature, log, and all of a sudden when they see it spike, they know they've ovulated. All right. I think I'm gonna go ahead and stop here. We're over halfway through this pack, so we still have a whole nother day, right? Not losing my mind. Yeah. So Wednesday, we'll finish this on Wednesday, and then we'll decide if Monday you want to do like review or you guys want to just have the day and study. Doesn't matter to me. All right. Enjoy the rest of your day. I'll see some of you in lab. If not, I will see you guys on Wednesday. Bye.